All right, we're going to get started here. Welcome everybody in person and online to our first public seminar of our 2024 series. Today I'm delighted to introduce Dr. Katrin Fleischer. Dr. Fleischer did her PhD in looking at nitrogen deposition effects on the terrestrial carbon cycle at the Free University of Amsterdam. She then went on to do postdoctoral research at the State University of Sao Paulo in Brazil and the Technical University of Munich in Germany, where she got involved in the Amazon FACE experiment project, and she specialized in tropical phosphorus cycle modeling. And then she went on to the Max Planck Institute for Biogeochemistry, where she worked on microbial modeling. What has really impressed me about Dr. Fleischer is her ability to take field-based experimental observations, thinking about really small-scale plant, root, soil interactions, and experimental responses of plants to elevated CO2 in tropical forests, and to confront global models that model process-based models that represent nutrients and plant strategies in a variety of ways to predict the consequences of these small-scale interactions for tropical forest ecosystems under global change. I think that ability to combine field knowledge with models and moving back and forth between them is a rare but invaluable trait for understanding ecosystems today and predicting how they'll change in the future. Dr. Fleischer is now an assistant professor at the Free University of Amsterdam, where she is a member of the Amsterdam Institute for Life and the Environment. So please join me in welcoming Dr. Fleischer.
Thank you, Sarah, for this really nice introduction. I'm really, really happy to be here and honored to speak to you today, to get the chance. And yeah, as Sarah said, I would like to use my time and tell you a little bit about my research and the research line that I've developed over the last years, and which I hope to continue in the future. So the title of my talk is Amazon Forest Responses in a Changing World. And you will see very shortly that I really focus strongly on those below ground processes, those really more difficult to observe but really critical processes in tropical forest for predicting global change effects in these critical ecosystems. So in case you got in late, just want to set the scene a little bit. These are recordings from our forest at the Amazon FACE site, where I've been working now since 8 years, since the start of my postdoc, and working in this project has allowed me to gain this model experimental knowledge, and has really shaped my career, has exposed me to different kinds of scientists, and has really shaped me into who I am today, and I continue along this path. So the Amazon rainforest, I'll switch off the music again, but just to give you a setting, Amazon rainforest is our largest intact global tropical forest in the world. It fulfills many, many critical processes for us. It's a large player of the carbon cycle. It stores immense amount of carbon. It is a substantial contribution to the terrestrial carbon sink. It also plays, apart from carbon, it plays a global role in the water cycle. So global climate, global carbon cycling is very much determined and controlled by this large rainforest. Next to this rather biogeochemical functions, it stores a vast amount of biodiversity, more than we have already discovered. We actually don't know the actual diversity level of this forest, but we think about 30% of vascular plants, vertebrates, and arthropods are found in this vast forest. So you see here a map that was from Hans ter Stege and collaborators in this really amazing report that has been published a couple of years ago now, The Amazon We Want. It is a really amazing resources for all kinds of things of the Amazon, for indigenous background, history, geology. So I really recommend, if you want to know more about the Amazon, to read this document. It's about how we go sustainably in the future. So I really use this as a nice resource. So I guess what I've told you about the Amazon, that is something probably most of you might have heard. Another thing I wanted to mention is this large rainfall generation that the Amazon has. So because of its vastness, it generates its own rainfall down windstream and actually generates all the water and the rainfall for the São Paulo region. So without that rainforest, the whole of South America would be a completely different system. So that, I guess, most of you have heard more or less. But one thing I want to talk more about today is about the soils and the underlying below ground processes that shape this forest, that allow it to function the way it does, and how this will control its response in the future. So when people first came to the Amazon, I guess no one really thought it would be poor soils, because you see this vast high productivity, and people were just astonished once they started looking into the soil and came across this conundrum, right? You have this very poor soil because the Amazon rainforest has been there for millions of years, so the soils had a really long time to be weathered. Having said that, it's not all of the soils that are really low in these rock-derived nutrients. So you see here a map of the underlying geology. It is quite heterogeneous. It's not all the same. It's not all Amazon forest is limited or on weathered soils, but you also have these areas here on the Andes where, due to erosion and uplift, you actually have new material coming in, and you can have quite some fertile soils. So this is a gradient of the phosphorus. So this is one nutrient I particularly focus on. It's a rock-derived nutrient and particularly important for plant growth. And this is one of the gradients here of the different soil types in the Amazon, from younger soils to more older soils. So you can see here there's first this large reservoir of phosphorus, but the total amount of phosphorus is being lost over millennia, over large amounts of time. And then about 60% of the area of the rainforest is these acrisols and ferrosols. And these are these deeply weathered soils. And there in this region here, like really low amount of total phosphorus. And a large portion of that phosphorus is what we call the residual fraction. So that is actually not very much plant available, not to say it's not available at all, but a large portion of this is not directly accessible by plants. And this organic fraction makes up what drives the system. So I want to tell you a little bit more about today how it's possible that we have this high productivity on these really low, poor soils. This is the map that we just generated recently with upscaling of field measurements of soil, and this is trying to give us some input for our modeling. So you see here these dark colors are low areas, low phosphorus, whereas the more lighter colors are some medium fertility of phosphorus, right? So how is it possible that these plants can be so productive on these low fertility soils? Well, I'll give it away. I think there are some self-sustaining mechanisms that have evolved over this millennia. Just as this self-sustaining rainfall gradient, we have this self-sustaining nutrient cycling. So these plants have evolved special mechanisms to deal with this low amount of nutrients, to retain it, and to recycle it very efficiently. So this picture I put in here is from a wood decomposition experiment at our Amazon face site. And what's really unique about the site, or at least I haven't seen it in other places, although I have not been to all these kinds of different places, but the fine roots come up to the wood, yeah? And you see standing dead trees, fine roots are coming up, getting into the litter layer. You have this really thick root mat, and that tells us that the plants are just right there, capturing all the nutrients that they can get out of the decomposing organic material, yeah? And that is quite unique. And that's not what we usually see in temperate systems or in more fertile systems. So one of the studies from Peter Witusek from 1984, I think this is one of the first ones that identified these mechanisms. You see here phosphorus in the litter fall, the total amount coming down, and the dry mass, so the concentration of this phosphorus in the leaves. And the line here are observations from temperate forest. And then this work showed us that in terms of nitrogen, at least for the sites they had, it was actually quite similar in terms of nitrogen cycling. But for phosphorus and also for calcium, the tropical sites differed quite a lot. So they retained a lot. There was a lot less nutrient return for a particular amount of of lithophore. So that told us something about these retention mechanisms and this recycling in tropical forest. And that set off a whole path of this general thinking that tropical forests are phosphorus limited. And of course, we're learning more that it's differentiated, and we want to understand more of what are the actual mechanisms and the functions these roots and others are doing. So now that is quite some time ago. Let me calculate, 40 years ago. And it's actually my birth year, so that's quite nice. So 40 years ago, and it took that long time until we had a first experiment in this region of the low basin of the Amazon, where they fertilized a mature patch of forest and proved for the first time that this forest is actually limited by phosphorus. So this is the AFEX site. This is near our Amazon face site. It is in this region of low phosphorus fertility, and they found after 2 years quite a substantial increase in productivity. The productivity was found so far in the leaves and in the roots, not in the stems yet, but that is something that could come up with later time. So I'm pointing this out here because we had fertilization experiment in different places, but these have pointed more towards nitrogen and phosphorus limitations, so more pointing towards this nuanced picture of not all tropical forests are the same. It really depends on where you are in the soil fertility gradient, and there is a large variety of function that we have to deal with. So this is in this area here in the basin, and this area is also associated with very slow growing trees, right? So they grow very slow. In my eyes, they're adapted to this low availability by just turning over slower and adapting to that amount that is becoming available by biological activity. So why does it all matter, and why have I even started thinking about the experiments? Well, I came more originally from a modeling world, and there this phosphorus limitation matters a whole lot for global predictions. So what we really want to know, while these fertilization experiments are telling us immensely, what we really want to know is how the forest responds with elevated CO2, because this is what the forest is facing. And the question is, can this forest capture more carbon? Can it grow more? Can it sequester more carbon? That is really the question. And if we're looking at experiments until now, so CO2 experiments until now have only been done with seedlings, and they have shown us that if you add fertilizer, which is here the gray bar, you get quite a substantial response of productivity to CO2. However, if you do not add fertilizer, this is reduced by 75%. These are seedlings, so you could say, okay, but they're in this strong growth phase. It's a good indication that phosphorus limits, but you cannot directly translate this now to a mature forest. However, models, you might be surprised, are actually quite late in accounting for this nitrogen and phosphorus limitation. So nitrogen has been put in these global models that are used for our climate predictions. They have been put in the 90s, I think, and only in the last decade or so, people have started to integrate phosphorus cycle in global scale, not because I don't think it's important, but because it is quite difficult to parameterize and understand all the functions. So what we've done here is extract all the biomass CO2 response in these global models that were used in the CMIP6. So that's great with the models. You can run a CO2 experiment and keep everything the same quite easily. And there you show there's one model that has a phosphorus. There's 5 of them now. I think it's 11 in total that have nitrogen. And you can see here that this phosphorus model, that one also agrees or at least also predicts a very, very strong limitation. So that matters a lot because our current predictions of remaining carbon budget, they all count with a really strong CO2 fertilization response. So in these, this is what we're using. So actually these forests, the tropical forests, they're assumed to buy us more time. They're assumed to capture a lot of the carbon that we're emitting. And then we have quite some reason to believe that's not the case. So in reality, climate change is probably going to accelerate and go faster than these models are currently predicting. A lot of other feedbacks are also going on, but this is why phosphorus matters a lot at global scale, and that in particular for the tropics. So there's a lot going on in the Amazon. There's deforestation, there's timber extraction, edge effects, fires, drought, and so on. And a lot of people are working on understanding these rather above ground processes, I would say. And what I want to highlight with here is that what is often forgotten is that all these plants have evolved on these poor soils. They have different strategies. They might grow slow, they might grow fast. And this all will affect how they regrow after deforestation, how they respond to a drought, how they respond to all these anthropogenic disturbances that we're exposing the forest to. And I think that is something that is rather overlooked, and is something that we need to integrate in better management and better projections for restoration and conservation, of course. So coming to my talk today, I separated the research in three topics. Firstly, I want to tell you more about what are these strategies for nutrient acquisition in tropical forest, and how do we expect those to control the tree's response to increasing CO2. So here I use a combination of models and observations to answer this, and that's quite vegetation focused. In the second part, I want to talk more about also how the soil microbes respond to these plant strategies under increasing CO2 and the consequences for the soil dynamic. Here I have a model across the gradient to explore these questions. And in the third step, I want to talk about what is the in situ evidence that we are starting to generate, how trees are adjusting their root strategies to changing conditions. So this is really from mature forest in the Amazon now, and here it's a combination of observation and experimental evidence. So firstly, let's go a little bit into the strategies that we're expecting to play a role in their expected response to elevated CO2. So here I can recommend you this review paper in New Phytologist that we published a couple of years ago, where we reviewed, to our understanding, the most critical or probably the most relevant strategies that forests and trees can use to acquire more phosphorus or to retain phosphorus more efficiently. So this is firstly the direct root uptake. So this is what everyone usually thinks of. You have a root and you take up nutrients from the soil solution. The other one, which is also a foraging strategy in our eyes, it's the arbuscular mycorrhizal symbiosis. So trees' roots associate to arbuscular mycorrhizae, and they extend their exploration in the soil, and also take up soluble or inorganic phosphorus. So these are foraging strategies for us. It's taking up the phosphorus that has been made available and it's in a plant available form. Then there are those two strategies where plants are actually mining for phosphorus. So they need to get phosphorus out of organic or an occluded form that is not available. And here we reviewed the acid phosphatase exudation. So phosphatase cleaves out phosphorus from organic matter without breaking off the carbon and the nitrogen out of this organic matter. So this is like a direct phosphorus acquisition pathway. And then there is this organic acid exudation, so they can exude acids which can lower the pH and potentially resorb phosphorus from minerals. So phosphorus is a very highly competed for nutrient in the soil. The minerals, the abiotic surfaces want to have it, the microbes want to have it, the mycorrhizae and the plants. It's never really just available, but it's always with someone, yeah? So these acids are supposed to liberate some of the phosphorus as well. And then we reviewed also this mechanism here of leaf phosphorus resorption. This is what I showed you from Vitusek's work, that plants resorb the phosphorus before the leaf is dropped to have a higher retention and be more efficient, to lose less, basically. And then we came up with this, with this concept here of how we think along this gradient that I showed you before of increased weathering. So here low phosphorus, high phosphorus. This is from this seminal work from Walker and Sayers. And we overlaid those mechanisms here, and we think that in this high, more fertile sites, we think that fine root foraging, just your normal way of doing it, would be pretty sufficient. And that is this, the lower you get, you will associate with arbuscular mycorrhizal to increase your efficiency. And these other mechanisms of resorption, exudation, of phosphatase, and the organic acid, that is costly. That doesn't come for free. Plants have to invest in these enzymes and making these specialized compounds. So this we expect to only happen when it's really needed, so at the more low fertile end. here on your right, you see the basis for the concept we draw. So this, we reviewed all the gradient studies and fertilization studies, and we looked at, okay, how is this mechanism changing? Is it predictable? Is it, in the case of fine root length, for example, it was quite predictable that it's increasing towards very low, so they become thinner and thinner and have to get more and more efficient. Also for phosphatase activity, it was a quite clear trend towards greater activity at the low end. But other indicators, such as the mycorrhizae, they went a little bit both ways, and it really depended on your initial status, so that's why we drew them in the middle. So while we had some evidence, you could see that it is actually quite scarce, and our knowledge, observational and experimental knowledge on this mechanism needs more work and need to have better observational grounds. So we think that CO2 would push our system further to the right. Yeah? So if you increase CO2, you increase production, trees need to take up more, basically deplete what is there and have to invest more and more in more costly things. So that's how we see the system. We think this will push to the right. Okay. Now I'll tell you a little bit about Amazon Face. This is this large-scale fertilization experiment that has been 10 years in the making now, and it's really built up now. So this is a real picture now from last year when I got to visit it. The towers are there, the cranes are there, and the experiment elevates CO2 in this ring. It's a system that has been developed in temperate system, but this is the first tropical one and faces a lot more logistical challenges, as you may imagine. So here just a couple more pictures. This is me in the crane looking down at the experiment, and this is here it's the kickoff event with the funders and students and journalists being there celebrating the event, and then a lot of pre-treatment data. So we've been going on pre-treatment data for 8 years, but now we need to do a lot more things now to have all the measurements we want before the experiment starts. So the experiment is a really, really large-scale, multidisciplinary experiment. It's not just carbon cycle people measuring the growth of the trees, but you have all sorts of people being involved there now. You have microbial biologists, of course, and soil scientists. We also have plant-herbivore interaction science going on. We are looking at the functional diversity of these trees, because none of these trees, hardly anyone has like a brother of their own species in our field, so everyone's alone, basically. And so we need to go this functional approach of trying to understand how is the individual tree functioning and why does it respond the way it does, and that will also allow us to upscale. We have this pre-treatment data of allocation of different components, and here you see that a large fraction of the biomass is in a very few number of trees, as you might have heard here. So that is also, of course, challenging to then capture these dynamics. Yeah. And when I started working there 8 years ago, they employed me to run a model ensemble, and that's what we did here. We got together a big group of experimentalists and modelers, and we took all the available data we had. We used 10 different models and asked the models what will happen if we elevate CO2 at the site. So again, we had models that had carbon, nitrogen, and carbon, nitrogen, phosphorus. In our case, we obviously gathered the most models we could with phosphorus. There were 6 of them at the time. And the result was as expected. Phosphorus would limit the biomass response. However, most interestingly, those models differed. You can see it's quite a large spread. Some models actually were able to gain biomass just as much as a nitrogen model perhaps, whereas others were quite rigid. And this set us off on this path of, okay, what made the difference, right? Why would some models be able to do it while other ones wouldn't? Well, it turns out that those phosphorus acquisition and phosphorus use mechanisms that I just told you about, that they made all the difference in these models, and depending on how rigid and how you parameterize that, you would get a different response. So we evaluated then those mechanisms that we had in those models. So not all of the ones that I told you about, have been actually put into models, but what we had at the time is we have a direct or indirect effect on growth. So phosphorus would down-regulate growth. That's the most obvious one. We have the shift in allocation, so they would invest more in roots to acquire more phosphorus when they needed it. We had a change in tissue stoichiometry for some models that allowed to have that stoichiometry flexible, that allowed them to grow more with less nutrients. And we had this phosphatase-based enzyme mineralization, which was important, and then a couple of models also accounted for this desorption from the soil matrix, and it actually gave quite a heterogeneous picture of which model allowed for which flexibility. Yes, and that I think I already made that point, but in these models, these mechanisms, they do come for free. So there's just phosphatases being upregulated if the plants are in stress, but it doesn't take any carbon away from the plant. The plants don't have invest or don't have an energy cost to do this. So this P acquisition at the moment in these models is for free. So this is why we set off to develop a new model, since none of these models is doing this cost now. So we developed here a module. It's a static module right now. It's not integrated in these dynamic models. But it tried to account for the cost of actually constructing an enzyme, for the cost of extracting an organic acid, and so on, and accounted for the fact that the lower your substrate is, the higher the cost will be. So if you're going with a particular enzyme for organic material, it's going to be, you probably have to invest more and more enzymes to get something out with a lowering substrate, yeah? Just to basically show that the efficiency decreases and a higher investment will have to make. So we put this in the model. And we used this gradient in the Amazon here we have the high P content now here on the right and the low P content here on the left, and you see that the total cost of phosphorus acquisition goes up with lowering phosphorus availability, and we use those for mechanisms that are usually, that are sometimes accounted for in models. Another interesting thing came out also is that this average cost per unit phosphorus acquisition goes up with lowering P, and that has to do with this functionality that we put in. So we think that the harder it will get for phosphorus to acquire, the more a greater share of their carbon plants will have to invest to get it. So the next step would be to integrate such a model then in these dynamic models. Yeah, we also calculated the amount of that cost compared to the amount of NPP, and you also see here that a larger fraction of the NPP would have to be invested. So there are two things basically. Sometimes you can have a lowering production, but a greater share of your production will have to go towards acquisition, and this is basically the expectation that we derive from here. This, I guess, has led me to be invited to this workshop last week, where Sarah, Michelle, Evan, and Jane were also part of the organizers. And I had the pleasure to be invited there and to be part of this workshop and combined it with my visit here at Kerry. And I just wanted to show you the things that we were thinking about there. So the workshop was about tropical soil fertility and root forest dynamics. We looked at how soil fertility affects production, but also mortality. And on the production side of things, we came up with this concept, thinking that on an axis of soil fertility, let's start here on the top, that you might see this effect of wood production on fertility, but we wanted to make the point there must be this hidden carbon cost of actually getting there. So if you were to actually plot the real NPP, because NPP is not just wood, but it's also all these investments plants have to make, this line would actually be more flatter. And at the same time, here we wanted to make the point that the wood production we're seeing is actually a result of these investments, that without these acquisition strategies, this line would actually be even sharper. Yeah, so this has implications for global change, for restoration, and so on. And we want to write a review concept paper on this dynamics, also in creating the mortality then. Let me just check that I'm sort of in the time. Yeah. Okay. So second section, what are the soil microbial responses? For this, I use the model Quincy JSM. This is a new dynamic vegetation model coupled to a microbial explicit soil model, and applied it across the gradient of the forest plots in the Amazon. And this is the gradient of these 30 sites that are modeled, and I model the historical runs, high CO2, no CO2, to see what are the more plant soil microbial dynamics of the system. This is the concept of the model. What I wanted to highlight here is you might think microbial explicit, why don't we always have microbes? Well, those models usually don't have microbes, and they model the function of the microbes implicitly, whereas these microbial explicit models actually have microbes as their own demand and entities, and they don't prescribe the stoichiometry of the soil pools, and you get actually like an emerging pattern of dynamics and biogeochemistry with depth, whereas before you'd have to prescribe stoichiometry and all these things. So you get this emergent thing, and that is really, really valuable, and you can do things like priming and things that go out of the sphere that we're usually in. So that's why I think we do need microbial models for modeling the future. So that's what we did here, and we applied it across the Amazon plots. What did we expect to happen? This is the simplified system, right? You have lithophile microbes, process everything, return it to the soil, to the plants. I'm sorry. And this system sort of is what drives everything, and the microbes drive the CO2 either being respired out of the soil or being formed into organic matter. So they're really at this critical point of diverting carbon and telling it what will happen with it. So I'm expecting that with plant phosphorus scarcity, we will see a lower lithophile with reduced quality due to the stoichiometry shifts I mentioned. I also expect that this whole cycle will slow down, that you have less and less phosphorus being circled around just because there's less available, plants hold on, and also the mineralization will slow down. And we have a higher root investment with lower phosphorus. The things of the costs and the exudates that I mentioned, they are not in this model yet, right? This is stuff for the future. So in the CO2 world, we kind of expect the same thing, right? We expect this lower lithophile, we expect a higher phosphatase-based mineralization, so these microbes would have to work harder to deal with this low-quality litter. And we expect, of course, that plants will sequester less carbon. It's quite an obvious one. But what will happen to the soil is really hard to predict because it can really go either way. Yeah, what's the deal with the soil? So I'll jump to the results. I show you here, every dot you see is one of those sites, and I always have them here along the x-axis of soil phosphorus. So you see, and the model is the darker and the observation is the lighter. So these are sites where we actually have measurements across these forest plots. You see the model agrees or like reproduces this reduction in wood growth with lowering fertility. It's at the range of the net growth, like the sink. It overestimates here the growth, but then it also overestimates the turnover, and that's why it sort of levels off at getting the right sink. And this is another topic we could have another day. So these are the results now only modeled, because these are things we don't actually have measurements for. So I walk you through some of the key results. Soil phosphorus limits the primary productivity, but actually not a lot. It was like a 10% reduction here. If you look at the numbers, actually not a lot. What actually happened a lot is an increase in fine root growth, so like almost a doubling of the productivity of the fine roots. And that, as a result, has a reduction in the plant carbon residence time. So the carbon in the plants cycle through much faster because litter produces, or she leaves the plant much earlier. So it had a speed up of the carbon cycle of the plants. We had a lowering litter quality and then also a lowering overall lithophile P. So my hypothesis of a slowdown of the cycle was confirmed here by the model. And interestingly, this microbial phosphorus acquisition did switch to this biochemical phosphatase-based pathway quite a lot. That was upregulated quite a lot. So they pulled out of the organic soil material, so this organic P cycle tightened. This is also kind of what we expected, but it was a quite neat thing to show. This is the first microbial model that includes nutrients, just to say that. So this is really first time we can model something like that. So here you see the CO2 effect, and now I split the sites in low and in high phosphorus. And again, I will walk you slowly through it. Firstly, CO2 increased the amount of carbon in plants and also in the microbes, so in living organisms. And also with that, of course, it pulled phosphorus. So there is more phosphorus stored in the living organisms with CO2. Due to this feedback of a faster turning of the plants to do higher litter production, the litter flux looked actually the same. So although they were less productive, they produced the same amount of litter, and that also caused that the soil carbon sequestration being at a much lower range than the plants, but that was quite comparable between the two sites because of this litter feedback. And the biochemical mineralization was upregulated even more, which led to a shift in the soil pools of phosphorus. So here in this low P site, the inorganic P didn't change because there was nothing left to be taken. At the low P site, they had to take it out of this mineral-associated organic pool, which is supposed to be a kind of a stable pool, so that is where it came out. Whereas the high site, that took up all the inorganic P that was there and then started to eat up, let's say, to feed into this organic matter. And that, I think, is quite the crucial point where what I want to make with this paper is that you basically need a shift of phosphorus from a pool that might also hit its limit in the future, right? This is only historical, and if I run this in the future, of course, I would run into some limit. Another interesting point from here is that the microbes in the plants, you saw that they take up more and more phosphorus. Overall, they're a really important reservoir of phosphorus. So the lower you are on this x-axis, the greater percentage of phosphorus that is actively cycling in the system is in the living biomass. So it's like 15% for the microbes and 9 to 10% for the plants. So in this low system, 25% of all the phosphorus that plants can use to live is in living biomass. And that, of course, you can imagine has implications for if you harvest these trees, if you take it out, if you disturb it, if you kill the microbiota for some reason and you lose this phosphorus, it will have consequences on your regrowth, right? Because phosphorus is not a nutrient that just can be replenished from the atmosphere or things. So it's an endless thing, and I think this is the second nice point that I want to make with this research. Here I forgot to mention this now. This organic cycle dominates the whole system in our model system, and it's 5 years that everything gets turned over. Yeah, so it's a very, very tight system there that has evolved. I mean, at least it's the model system, but we have good indication to think that this is true in the real world. Okay, I sped up a little bit in the second part, but now that gives me good time to talk about the third part, which is a really exciting part. And that is about what is the in situ evidence right now, because you could say you could be a model skeptic and think, yeah, but how do we know that they're really adapting, and you only showed us young trees and I don't know, I'm not buying it yet. So I'll show you something to convince you a little bit more that trees are able to adjust their root strategies in changing conditions. So while we had this long phase of pre-treatment data at Amazon Face, one of the activities we did is construct an open-top chamber experiment where we elevated CO2 for understory plants. So they're about that size, that thin, and in this system we tested all of our above ground and below ground measurements, and we wanted to know what happened. So I will show you some results from this OTC experiment, which was running for 2 years. And this is the result from the above ground component of the OTC, which just last week was published in Plant Cell and Environment. We are really super proud. This is our first real CO2 in the Amazon research. So we saw quite a lot, right? Some people were like, it's only understory. The CO2 concentration in the understory is quite high anyway because the CO2 builds up overnight. So some of us were thinking, well, if you add 200 ppm now, what's it going to do? It sees that anyway. But to our positive surprise, quite a lot happened. So you saw an increase in photosynthesis assimilation rates. You saw an increase in the water use efficiency of these plants, and an increase in stem diameter. So you see first here the growth rate for the different diameter classes, and there was a significant effect for ambient and elevated. But of course, it matters of how large you are already. So they calculated also the relative growth rate, and you also see an effect there. So these plants were growing more in this really low fertile soils, although they were in the understory. So that gave us obviously quite some good motivation for the big experiment. But even more exciting, but don't tell this our above ground team, even more exciting is what happened below ground. That was really mind-boggling. We knew that the litter layer was important. We knew this just because we saw the roots in the litter layer. So what we had done there with the below ground is really separate the topsoil and the litter layer effect. You see here on the right the response of root productivity, root morphology, and enzyme activity and mycorrhizae. To summarize, in the litter layer, which is here the red one, the roots became thinner. You had a higher phosphatase activity, so also thin roots are associated to high enzyme activity, so they just became thinner and seemed to invest in biochemical mineralization of organic P, the same mechanism we talked about, and we called this do-it-yourself. So they went in the litter and worked even harder to get material out of the decomposing litter, whereas in the topsoil, the roots actually became shorter. There was a reduction in root productivity. They pulled out. They didn't make as many roots as they had before, and they invested more in mycorrhizae. So in the topsoil, there was this outsourcing mechanism. So they did two completely different things in like a separation of like 10 centimeter in this 2 meter diameter little cage. Yeah, I thought that was really mind-boggling to me as a modeler, that you can see this effect. So this is the diagram we came up with. It's still work in progress of what's all happening. But another really interesting thing is this here in the soil layer. Again, in this really small patch, they cored 3 times after 2 years, cored 3 times and saw a reduction in the organic phosphorus in the topsoil. So that told us, there we didn't see the enzyme, but that told us just this less organic material from the litter was basically trickling down or being converted in this soil, right? So this is another indication that it was pulling out of this organic. And when I saw this, I was like, and I had the same result in my model, I said, oh my God, you guys, we are onto something, right? So it's always nice when there is some agreement between the model and the observation. So that's what's coming out. That's work in progress. Hopefully you see this published very soon. So in a summary from the OTC, again, the below ground, the total fine root productivity did not really increase as the model predicted, but they rather invested in other mechanisms. However, the amount of investment this mechanism costed, of course, we don't really know. We cannot really directly measure that. However, that's a sort of a contradiction to what our current models are doing. There is a large plasticity in root morphology and in these mechanisms in the different litter in the soil layer, which is really, really, really cool, but also really important that we get a handle of this in models because they're really static in that way now. And we had this greater investment in organic phosphorus mineralization and a shift of soil organic into the plant biomass. Yeah, we have good reason to think that it didn't went into the microbes, but we have reason to think that plants actually acquired this phosphorus. So now again, you could say, oh, but these are just small plants. How about the mature forest? Well, I have a little bit of evidence also that they are also plastic. And here, this is a study also from our Amazon face site of different root cores over a period of 3 years to capture the seasonal dynamic of the roots. So here we looked at the morphology. And one interesting thing that came out, you see specific root length, diameter, and density. And here on the x-axis, you have accumulated precipitation of the last 6 months. So you're seeing this shift in thinner and less denser roots after rainfall periods and the other way around. And again, we saw that thinner roots are associated to phosphatase. So you could think this is just water, but I think it's both. It's both coming together, that they're basically, once the environment is good and there's material trickling through and the environment has organic material, they invest in these thin acquiring roots. So that was also a really, really cool thing to come out. And these are cores in the mature forest. So you don't know which root you got, but it is the community response. And the other thing is work from the AFEX side, so from this fertilization experiment side that you saw earlier. And there fertilized large patches with phosphorus and cations, as you heard. And we did the in situ root exudation. That's also a really tricky thing to do. You have to excavate the root, do like a pipette, wait a long time. It's a really tricky measurement to make. And this was the first root exudation measurements in the Amazon. And we found here a treatment effect. So in the control plots, let's start on the right. This is a total amount of carbon that's been exuded, and that was less in a phosphorus addition plot. So that tells us, okay, they invested less in this pathway once the phosphorus was there. So that tells us, okay, that exudation, whatever they're exuding must have something to do with phosphorus. And here, this is the organic acids. This is what we actually wanted to look at because we know this matters for phosphorus. And there was also an effect. I put them next to each other to show you the difference in scale here. So this is a log scale, but this is in the thousands, and this is in the 100. And all the other carbon that came out from, I'm hearing at least from my experimental friends, we don't really know what their function is and what their role is. It has large amount of nitrogen also coming out. So lots of different functions surely this exudates are fulfilling that we're just starting to understand. I'm getting to the end. I have one last slide just to show you that while we have all this evidence, and I wanted to make the point that we are getting there and we're seeing changes. However, if you look at the data for root traits here, and you see here the color for the different biomes. So the red color is tropical forest, and these are the root functional traits. So you see it's like overall we know very little about root functions, and tropical forests have even less available of data than at global scale. So we need a lot more investment in understanding these traits and functions. Then, of course, it's not just about nutrients. This is from a review we did with the TropiRoot group where Sarah and I am part of, where we try to see how root biomass, turnover, and investment changes across two gradients of dry and wet and fertile and infertile. So that's something to take account in the future. It's obviously not just phosphorus, but it's a combination. Plants need to acquire both things from the soil environment. And it doesn't stop with the roots, right? The roots are connected to the whole plant growth strategy, and it will feed back into all these above ground things that we know more about, and we need to be able to associate those functions to particular strategies above ground. Yeah? Just wanted to make the point that this is even more trickier to then make this whole plant perspective of growth and function, but this is what we need for models, this is what we need for our understanding and management practices. And yeah, this is where we hopefully can make a difference in the future. So this is what I had to say. This is a picture from our TropiRoot network where I actually met Sarah first time in person last year, and this is what kicked us off working together. So yeah, stay tuned for more on this model experimental integration for more below ground functions in global models. Thank you very much.
All right, thank you for Katrin for a wonderful talk. If we'll take questions now, and Katrin, if you could repeat the question from our in-person audience so that the online audience can hear. Yes.
I go with yes.
Thanks, Katrin, for a really cool talk. When you were talking about different mechanisms plants can use to acquire phosphorus in your first part of the talk, I wonder, does like just root respiration and production of CO2 and carbonic acid, does that play any role at all in weathering of P minerals, or is that not really a thing?
The question was about whether root respiration plays a role. I've not heard that. I don't want to exclude it, but I've not heard that this is something. There's weathering, but I've thought it's more those other kind of acids. But it could be that this is, I mean, in this kind of system, weathering doesn't play a role anymore because there is no more weatherable minerals. But perhaps in a more fertile site or more rock apatite. But I haven't heard that, but you're welcome. Evan?
Yeah, really interesting work, I think. I was curious, so when you looked across the rainforest sites and that fertility gradient,
Yeah, so Evan asked whether this nonlinear shape we saw with soil fertility of productivity, right? What could be driving this?
Yeah, it was a number of different variables.
Yeah, it was a number of different variables. Yeah, I've actually, I'm also very intrigued about this threshold. A lot of observational data and also the model seems to be showing some cutoff where sort of phosphorus limitation becomes more important. I do see it as the point of where perhaps that amount of phosphorus is needed, and that at higher phosphorus other things might be starting to be limiting. So that maybe then the cation limitation comes in, or light, or other dynamics. So I see it more that really in this low range you have like a linear response, and that at some point it does level off. So I also believe there's a threshold. I haven't thought a lot about the mycorrhizae and how that changes across. It could be that perhaps, yeah, we're thinking mycorrhizae play a role in this medium range a lot. And we actually thought about that a lot yesterday. Maybe we need to talk more about where we think the mycorrhizae and what kind of function they play along this range. Thanks. Jane?
This was really fun to see. Thank you. I'm really curious, though. You're doing the hard work of collecting those exudates off those roots, which
My collaborators did, to be honest.
not fun. So you see differences in the carbon, but are you seeing compositional, and are you looking at what the diversity of that is? And do you have a sense that the trees are trying to get rid of carbon, or is there, like, can you tell anything compositionally what's happening in those exudates?
Yeah, thanks. So Jane asked whether the composition of the exudates, whether we know more about the compositional change, and whether maybe trees are just getting rid of carbon. This is a really interesting question that people get really upset about. So to the first part, the composition, so the study was about organic acids. So Tatjana Reichert looked at the different acids, so malic acid, citric acid, and so on, and we saw a compositional shift. I can show you the results for that. However, the analysis at that point was limited to that. And she measured the total carbon and the total nitrogen, and we have quite a high amount of nitrogen in these exudates, which I thought was strange. But the organic acid made a small fraction, and the rest, at least at that point, we don't know. I don't know if we can repeat and look more into if we know what we're looking for. As to the point of whether they're just getting rid of, I don't exclude that hypothesis. I do think that there's a pathway of plants that could get rid of carbon. I do, however, think that, yeah, they have probably evolved different, like we know their strategies and we know they have functions, these kind of compounds that they exude. So I'm not on this team of, oh, it's just a surplus, right? So I think the truth is somewhere in the middle. But I mean, interestingly, there's the Uke phase experiment. There's a phase experiment in Australia that did see a large effect of just carbon flowing out. So you had a small effect of photosynthesis and a high increase in soil respiration. So that experiment actually made me think, oh, maybe it is just, if they can't get any more, then maybe it just really does flow through. I don't exclude it. I think it's somewhere in between. Yeah.
All right, we have a couple of questions online. So one from Wangguang Tang. He has two questions. He said, is it really so costly for phosphorus uptake? Do you think trees can store phosphorus or have luxury consumption when phosphorus is rich in the environment? And then the second question is, in the open-top chamber experiments, does the temperature increase inside the chambers?
Okay, thank you. Phosphorus acquisition, well, yes, there is luxury consumption of phosphorus, and experiments, Aqua Salud from Sarah showed me that is really the case. They have, and we have more and more evidence. Last week at the workshop, we've seen research of large amounts of nutrients can be stored in the wood. What it's doing there, we don't really know. We don't know how good they are at storing it or getting it out. So yes, there's definitely this storage part. But I still think it's sort of costly to get because it's never really, really richly available around their roots at any one time. Yeah, because as I said, everyone is competing for phosphorus, the microbes, the minerals, so they never have a flush of phosphorus like you would have in a fertilization experiment. So I do think like of getting it, there would have to be an investment, But yes, there is the storage and this retention component which can play a role. And to the OTCs, yes, it gets warmer in there. You don't want to be a long time in there, but there's a fan, and they're doing different things to basically compensate for the changing in the climate there. We'll have to ask our above ground people, but they are obviously thinking about this. And there's a fan, and they have also done something with the water, because it excludes a little bit of the rainfall. But yes, it's a chamber, but yeah, I mean there's control chambers to look at that. So yes.
So Tristan Tabor is asking, I know there's been some studies dealing with biomorphic changes in plants causing unintended consequences. For example, growth rates increase with more CO2, but then the trees may have less structural integrity or superficial nutrient application changes, root pathways, and trees then may be more likely to experience windfall events. Mm-hmm. Do you have any insights into the changes in morphology and potential impacts with the changes you're looking at?
No. No. That's interesting, and I guess this is something we tried to think about more last week at this workshop, because honestly, yes, so far we've been thinking CO2 speeds up production, and we're aware of this potential trade-off with mortality, but now I'm learning a lot more from Evan and colleagues that is not so simple, and that we can expect some feedbacks between fertility and mortality also. And yes, we think that if you don't invest so much nutrients in your tissue, there must be some drawback of that, right? You might not be investing so much in defenses, or as the question pointed out, that you might not have this structural implication. But yeah, I think we're really, really limited in our understanding there, and I don't know. Something to think about, those secondary effects then, yeah.
I'll ask a question. So it was really cool to see the whole plan for the Amazon face experiment, and I'm really curious about the herbivory aspect of it. Oh, yeah. I'm curious, do you know what is being found on herbivory effects on CO2? And then also, we found that and different studies have found that herbivory has a major causes a major loss of carbon from trees in tropical forests. Is there a need to model herbivory in these global change models?
Firstly, to the herbivory and the CO2. So I was part of this team that proposed and put this together. So I've learned a little bit about this, that we basically expect with lowering quality, that with higher CO2 the leaves will be less nutritious, and that make them theoretically less palatable and less attractive for herbivores. And I know that they're installing things to look at this now, and there's obviously this dynamic, right? You only have that patch, and a herbivore that can move will just go to a different tree, and then there are the herbivores which cannot move. But that's as far as it goes from me for the hypotheses. UGFACE has looked at FRUS, so they have measured that component. I don't know now whether there's a CO2 effect, but we could look at that. And whether we need to model it comes down anyway, doesn't it? One way or the other. No, I think it matters a lot, because if you only have the plant material coming down, microbes will have to convert it, whereas whether an herbivore eats it and the feces come out the other end, then it's actually plant available things. So it's called circumvents this mechanism. That much I know, but I don't know any model that has tried to look at this effect then, and whether so far this is being ignored, and the microbes have to do the job of the herbivores. Yeah.
Thanks. Any other questions in the audience here? Otherwise, we have one more question online, which is, what are the perceptions of indigenous people and other local people about the changing Amazon with global change and as CO2 levels rise, and then how forests might adapt in the future?
Oh yeah, that is a really good aspect to mention. So we have this social environmental team they used to be socio-economic, now it's socio-environmental. So these are people that are exactly thinking about this. So they do studies of doing surveys, going out to the indigenous community and asking them exactly that question. This is ongoing. I don't have like a feedback, but these people are perceiving that their forests are changing. If you followed the news, the drought in the Amazon last year was as severe as never before. People are like, I've never experienced that. The Rio Negro was completely dry. And also the experience of the dryness of the air. People said, I live here 12 years, I've never had it so hot. So it's this hot and dry air, the VPD that is changing, and the burning of the forest. So they are surely perceiving that their forest is changing. But I don't think they quite know that it's people that have done it. Right, so they know it's changing, but I don't think they know that it's us that have done that. And so we're aware of this impact, and of course the experiment will blow more CO2 in the air, right? So this is a delicate thing. So we're trying to work with the community. There are some of us that go out on visit these communities, have informational sessions, have like knowledge stands, and we try to be in contact. And other experiments in the Amazon have like paved the way for that. So we are aware of this. And we want to be in touch with these communities. And another interesting aspect is that they're looking at the use of the trees in our patch. So we want to identify which trees have human use and pay a particular focus on them as another functional trait, basically, because this can be relevant then for agriculture, for adapting and management strategies. So we're paying attention to this human use of these trees.
Great, well, I think we'll end there. For those of you here on campus, if you don't have a meeting with Dr. Fleischer this afternoon, we're having lunch now in the lunchroom. We're also having a happy hour this afternoon, and people can come over to my house for dinner tonight, so just let me know if you want to join. But please join me in thanking Dr. Fleischer for a wonderful seminar.
Thank you, guys. Thank you.